Zinc as a gatekeeper of immune function. Published by PubMed Central. Abstract. After the discovery of zinc deficiency in the 1960s, it soon became clear that zinc is essential for the function of the immune system. Zinc ions are involved in regulating intracellular signaling pathways in innate and adaptive immune cells. Zinc homeostasis is largely controlled via the expression and action of zinc importers, ZIP1 to 14, zinc exporters, ZNT1 to 10, and zinc binding proteins. Anti-inflammatory and antioxidant properties of zinc have long been documented. However, underlying mechanisms are still not entirely clear. Here, we report molecular mechanisms underlying the development of a pro-inflammatory phenotype during zinc deficiency. Furthermore, we describe links between altered zinc homeostasis and disease development. Consequently, the benefits of zinc supplementation for a malfunctioning immune system become clear. This article will focus on underlying mechanisms responsible for the regulation of cellular signaling by alterations in zinc homeostasis. Effects of fast zinc flux, intermediate zinc waves, and late homeostatic zinc signals will be discriminated. Description of zinc homeostasis-related effects on the activation of key signaling molecules, as well as on epigenetic modifications, are included to emphasize the role of zinc as a gatekeeper of immune function. Keywords: zinc flux, zinc wave, homeostatic zinc signal, immune function, zinc deficiency, signaling pathways. 1. Introduction. The transition metal zinc has long been known to be essential for the growth of fungi and development in rodents. 1, 2. Symptoms arising from zinc deficiency in animal studies were reported to be growth failure, loss of hair, testicular atrophy, as well as thickening and hyperkeratinization of the epidermis. Despite these observations, it took a century until zinc was accepted to be essential for human beings. 3. Previously, zinc deficiency was considered to be rare. Nowadays, however, zinc deficiency is known to be very common, especially in developing countries. 4. Worldwide, about 2 billion people are estimated to be affected by zinc deficiency. In developing countries, zinc deficiency is the 5th leading cause for the loss of healthy life years. In industrial countries, mainly the elderly population is affected by zinc deficiency. 5. Nearly 30% of the elderly population is considered to be zinc deficient. Since zinc homeostasis is known to be important in immunological reactions such as the inflammatory response and the oxidative stress response, multiple chronic diseases observed in the elderly are probably related to zinc deficiency. Hence, diseases such as rheumatoid arthritis, diabetes, atherosclerosis, impaired cognitive function, as well as age-related macular degeneration, AMD, may be due to zinc deficiency, worsening chronic inflammation and triggering oxidative stress. 6, 7, 8, 9. Besides the elderly, vegetarians or vegans and patients suffering from renal insufficiency or chronic diarrhea are affected by zinc deficiency. 6, 9, 10, 11, 12. In humans, clinical manifestations of zinc deficiency are similar to those observed in rodents and include weight loss, growth retardation, atrophy, and immune dysfunction, as well as increased oxidative stress and a boosted inflammatory immune response. 3, 13, 14, 15, 16. Dietary insufficiency and/or compromised uptake can result in perturbation of zinc homeostasis in humans. 12, 17, 18. Besides zinc, vitamins and other transition metals such as copper and iron, as well as calcium, are essential for adequate immune function. Deficiency or exceeding levels of those nutrients can be associated with the occurrence of distinct diseases. In this regard, a multitude of diseases such as rheumatoid arthritis, 19, multiple sclerosis, 20, 21, 22, impaired cognitive function as seen in Alzheimer's disease, 7, 23, and AMD, 24, were reported to go along with skewed concentrations of the above-mentioned nutrients. 2. Zinc metabolism and homeostasis. At 2 to 3 grams in total, zinc is the second most abundant metal in humans and is distributed unequally throughout different organs and tissues. Prostate, pancreas, and bone are considerably high in zinc, containing up to 200 micrograms per gram. In contrast, zinc concentrations in heart, brain and plasma are comparatively low at 1 to 23 micrograms per gram. Although plasma has only 1 microgram per gram, it is probably the most important reservoir for zinc homeostasis. 5. Red meat and oysters are rich dietary sources of zinc. 5. Foods prepared from unrefined cereals, legumes, or plant parts rich in phytates decrease zinc bioavailability by binding zinc very effectively. 25. Zinc is resorbed in the intestine by specific zinc-transporting proteins and distributed in the human body. In serum, free zinc is rare since it is largely bound to proteins such as albumin, alpha-2 macroglobulin, A2M, and transferrin. Albumin binds zinc with a relatively low affinity, A2M with a medium affinity, and transferrin with a high affinity. Subcellularly, zinc is distributed between specific zinc-storing vesicles called zincosomes, approximately 50%, the nucleus, approximately 30 to 40%, and the remainder is distributed between the cytoplasm and other organelles, 26-27. In the cytoplasm, zinc is largely bound by zinc chelating proteins called metallothioneins, MTs, discovered in 1957, 28. MTs play an important role in zinc homeostasis by complexing about 20% of intracellular zinc. MT1, for example, binds up to 7 zinc ions, making it specifically suitable to function as a zinc buffer. 29. During evolution, cells not only evolved metal-binding proteins to maintain low cytosolic metal ion concentrations to protect themselves against high cytotoxic metal concentrations, but also mechanisms of zinc compartmentalization and zinc sequestration. Modulation of available cytosolic free zinc is facilitated by zinc storage in zincosomes or other organelles such as endoplasmic reticulum, or Golgi apparatus respectively. 30. Zinc distribution between the cytosol and organelles is mediated by specific zinc-transporting proteins, such as zinc importers or zinc exporters, or via membrane channels. 29-31. The cytosolic free zinc ion concentration is maintained at remarkably low picomolar to nanomolar concentrations. 32. However, through zinc release from protein and organelles, zinc concentrations can increase transiently and locally, enabling zinc to affect gene expression, enzymatic activity, and cell signaling. 31-33. Zinc homeostasis is particularly controlled by 24 zinc-transporting proteins, and 4 MTs, Montana 1-4, playing coordinated roles in the distribution, transport, and maintenance of intracellular zinc level. MT1 and MT2 are expressed ubiquitously throughout the human body, play an important role in liver and kidney function, and are inducible by various stimuli such as metal ions, cytokines, glucocorticoids, and oxidative stress, respectively. In contrast, MT3 and MT4 are expressed cell-specific. MT3 is primarily expressed in the central nervous system, whereas MT4 is primarily found in epithelial tissue. The expression of both MT3 and MT4 is very strictly controlled, highlighting distinct functions of MT isoforms in different organs and tissues. 34, 35, 36, 37. Based on their membrane topology, zinc transporters are divided into 2 major families. 1,4 zinc transporters of the SLC39's family, ZRT, zinc-regulated transporter, like IRT, iron-regulated transporter, like proteins, ZIP1-14, that increase cytoplasmic zinc, and 2,10 zinc transporters, ZNT1-10, of the SLC30's family that lower cytoplasmic zinc by transporting zinc either out of the cell or into intracellular organelles. 38. Since zinc transporters are found in a myriad of different cell types in the human body, both on the outer plasma membrane and on organelle membranes, such as mitochondria, Golgi apparatus, lysosomes, and the necessity of a proper zinc level for a specific cellular function is emphasized. 38-39. Moreover, zinc distribution was more recently mentioned in relation to receptors and ion channels other than ZIPs or ZNT, for example voltage-dependent calcium channels, transient receptor potential channels, nicotinic acetylcholine receptors, and glutamatergic receptors. Additionally, diffusion of zinc bound to amino acids is known. However, these mechanisms need to be investigated more in depth. 39. Interestingly, zinc dependence was found in all classes of enzymes, i.e., transferases, hydrolases, lyases, isomerases, oxidoreductases, and ligases. 40-41. The physiological importance of zinc was moreover supported by in silico studies which showed that about 10% of the overall human proteome can potentially bind zinc. Zinc binding can be facilitated by a variety of binding motifs including 1. Zinc finger motifs, 2. Really interesting new gene, ring, finger domains, 3. Limb domains composed of 2 adjacent zinc finger domains, and 4. Plant homeodomain, PhD, domains similar to ring fingers, 42. Thus, interaction of the transition metal zinc with proteins is highly complex. The zinc proteome consists of 2 major groups, comprising enzymes and transcription factors respectively. Those cover 90% of the overall zinc proteome, indicating that zinc is involved in regulating catalysis and transcription, 43, 44. Because zinc is required for catalysis by some enzymes, as well as for maintaining enzyme structure, 26. Intracellular signaling pathways can be altered due to altered zinc availability, affecting cellular maturation, differentiation, and function, 45. 3. Immune function during zinc deficiency. The importance of zinc for proper immune function is best observed in zinc-deficient individuals. Zinc deficiency has been known for 50 years, 15, and is associated with skin abnormalities, hypogonadism, cognitive impairment, growth retardation, and imbalanced immune reactions which favor allergies and autoimmune diseases, 3, 5. In the case of inherited malfunction of zinc homeostasis, as seen in acrodermatitis enteropathica, zinc deficiency can be lethal, 17. Zinc deficiency can be classified by severity and is divided into severe or marginal zinc deficiency, respectively. Severe zinc deficiency is often observed because of malfunction of zinc uptake in the intestine. This is reported in patients suffering from chronic diarrhea, patients being treated with penicillamine, patients receiving parenteral nutrition without zinc, or following excessive alcohol consumption, 46, 47, 48, 49. Patients present clinical symptoms such as lymphopenia, decreased ratios of T helper, Th, cells to cytotoxic T cells, decreased natural killer, NK, cell activity, and increased monocyte cytotoxicity. The most severe form of zinc deficiency observed in acrodermatitis enteropathica. This zinc malabsorption syndrome is inherited as an autosomal recessive condition and is due to a mutation of the intestinal zinc uptake protein ZIP4, 1750. Acrodermatitis enteropathica is characterized mainly by diarrhea, weight loss, recurrent viral and bacterial infections, dermatitis, hair loss, and neuropsychological disturbances, 51. However, all observed symptoms can be corrected by high dose, 1 mg per kilogram, zinc supplementation, 52. Other zinc transporter-related diseases are discussed later in detail. Marginal zinc deficiency is characterized by slight weight loss, rough skin, oligospermia, and hyperammonemia, 47. It is probably caused by nutritional zinc deficiency, often seen in vegetarians or vegans, due to the consumption of high levels of zinc chelating agents in food originating from cereals, legumes, or plant parts. In these foods, lignin and phytates counteract zinc absorption by binding zinc and reducing its bioavailability, 1053. Nutritionally related marginal zinc deficiency is prevalent in the elderly population. Hence, a correlation between impaired immune function and zinc status is likely in older people, 654, 55. Within the 7th decade of life, the human immune system undergoes dramatic age-related changes, termed immunosenescence. Associated with this condition is an increased incidence of inflammatory disease, most notably cardiovascular diseases, whereas the immunological response to vaccines is typically impaired, 56. The underlying zinc-modulated molecular mechanisms and signaling pathways are discussed below. The overall frequency of zinc deficiency worldwide is estimated to be higher than 20%, 18. Interestingly, zinc supplementation is already widely practiced and approved for clinical treatment of multiple diseases. Zinc has proven to be very effective for the treatment of pediatric diarrhea, saving millions of children's lives in developing countries such as India, 48. The Food and Drug Administration, FDA, approved zinc supplementation for the treatment of Wilson's disease, a genetic disorder in which copper builds up in the human body, 57. In the elderly population, age-related macular degeneration, AMD, is of frequent occurrence in AMD progress, which can result in blindness, can be treated successfully by zinc supplementation, 58-59. Furthermore, not only the very young or elderly benefit from zinc supplementation, as shown by studies with patients suffering from 1. Viral infections, e.g., the common cold, diarrhea, chronic hepatitis C, or human immunodeficiency virus, HIV. 2. Bacterial infections such as shigellosis or Helicobacter pylori. 3. Parasitic infestations such as acute cutaneous leishmaniasis or malaria. 4. Autoimmune diseases such as type 1 diabetes mellitus, T1DM, and rheumatoid arthritis. And 5. Transplant rejections, 60. This widespread variety of clinical manifestations makes zinc deficiency a serious nutritional problem. However, to date, no reliable biomarker to assess zinc status exists, 61. Thus, zinc deficiency is difficult to diagnose. Overall, zinc contributes to the overall regulation of immune cell function, influencing several signaling pathways. Hereby, zinc acts in a direct manner by binding reversibly to regulatory sites in signaling proteins, or indirectly by influencing enzymes such as phosphatases which are a component of and regulate signaling pathways. 45. Zinc homeostasis is essential for multiple aspects of the immune system including hematopoiesis, cell maturation and cell differentiation, cell cycle progression, and for the proper function of immune cells. 62. During inflammation, adequate zinc status is essential since in acute phase responses zinc is transiently transferred from serum into organs, especially the liver, causing transient serum hypozinsemia. This transient loss of serum zinc is eventually rebalanced during resolution of the inflammatory response. Here, zinc is probably released from tissue into serum. One proposed reason for this complex mechanism is to act as a danger signal for immune cells. 63. Since extracellular microorganisms are also dependent on zinc availability, zinc sequestration by the human immune system helps to combat invading pathogens. This is facilitated due to expression of pro-inflammatory acute phase proteins including interleukin, IL-6, which upregulates expression of zinc-binding peptides such as MTs and A2M. 64. In immune cells, on the one hand, increased intracellular zinc levels can intoxicate engulfed pathogens and act cytoprotectively by neutralizing reactive oxygen species, ROS, and nitrogen species, RNS. In general, zinc homeostasis and zinc signals are crucial to counteract inflammatory diseases, and the correlation of undernourishment with severe inflammatory diseases is accompanied by prolonged and severe forms of serum hypozinsemia. In the literature, it has been suggested that hypozinsemia goes along with elevated inflammatory mediators, e.g., ROS, and antimicrobial peptides such as calprotectin or matrix metalloproteases, MMP, causing tissue injury, especially in liver, lung, and spleen. 63, 65, 66. In general, cellular function, such as the intracellular killing of harmful pathogens, cytokine production as well as ROS production, are dependent on zinc and are impaired due to zinc deficiency. Zinc deficiency also adversely affects the maturation and function of T and B cells, which occurs through dysregulation of basic biological functions at the cellular level, described later in this review in more detail. For T cells, a disturbed ratio of Th1 and Th2 cells in favor of Th2-driven allergic reactions is a well-known consequence of zinc deficiency. 67. Zinc flux and homeostatic zinc signals, as defined below, are highly important for adequate T-cell differentiation, and this observed malfunction can be reversed by zinc supplementation. 68, 69, 70, 71. Moreover, the pro-tolerogenic immunoreaction is triggered by long-lasting changes in intracellular zinc levels due to induction of regulatory T cells, TREG, cells and dampening of pro-inflammatory Th17 and Th9 cells. 72, 73, 74, 75. T-cell development is strongly dependent on DC activation. Interestingly, zinc signals were recently shown to induce a tolerogenic DC phenotype in vitro and in vivo. Herein, zinc suppressed MHC2 expression and enhanced programmed cell death 1 ligand 1, PDL1, and PDL2 expression resulting in the manipulation of the TREG-Th17 balance in favor of TREG cell development. 76. Moreover, on the molecular level, Zinc inhibits the IL-6-induced STAT3 signaling cascade essential for Th17 development, 74. Another potential target of zinc-mediated Th17 manipulation might be found on the epigenetic level, since several epigenetic enzymes as acetylases and methylases are regulated in a zinc-dependent manner, 72, 77. A malfunctioning adaptive immune system has been observed in the elderly population, whereby secretion of pro-inflammatory Illinois-6 is pathologically elevated, while T-cell activation was reduced, as were responses to stimulation or vaccination, 6, 56, 68, 70, 78, 79. Interestingly, all these pathologies can be improved due to zinc supplementation, highlighting the significance of zinc for a well-balanced immunoreaction. Regarding innate immunity, zinc deficiency leads to prioritization of maturation of innate immune cells such as monocytes. In this case, differentiation was promoted by the reduction in the concentration of intracellular free zinc, 80. This was facilitated by induced expression of the zinc-binding heterodimeric protein calprotectin, 81. In general, calprotectin was also highly expressed in neutrophils, 81, 82. Recently, zinc deficiency has been shown to negatively influence critical neutrophil functions such as phagocytosis, oxidative burst, degranulation, cytokine production, chemotaxis, and neutrophil extracellular trap, NET, formation, 83, 84. These observations are in line with an impaired ROS production during zinc deficiency, which is needed for NET formation, and for intracellular killing of phagocytosed pathogens by neutrophils, 85, 86. Furthermore, NK cell function was weakened when zinc signals are absent. In this regard, recognition of major histocompatibility complex class 1, MHCI, on target cells, as well as lytic activity, was decreased, 87, 88, 89, 90. In contrast, zinc supplementation increased differentiation of CD341 progenitors toward NK cells and their cytotoxic activity, 91, as well as NK killing activity, and intracellular perforin concentrations, 90. Besides cellular immune responses, zinc is also indispensable for proper RNA transcription, DNA synthesis, as well as cell survival, 27, 92. In relation to cell survival and apoptosis, adequate intracellular zinc levels are needed because apoptosis is triggered by zinc deficiency. Furthermore, cytokine function and secretion are adversely affected by zinc deficiency, impairing the function of the basic messengers of the immune system. Thus, zinc is crucial for the appropriate development and function of the whole immune system, including innate as well as adaptive immunity, and the affected signaling cascades and networks are described in detail below. 4. Types of zinc signals. As indicated earlier, intracellular as well as extracellular zinc is usually bound to protein. From protein, zinc can readily be made available but also made unavailable. About 10% of total human proteins bind some zinc throughout the lifetime of the protein, as it is essential for catalytic or structural functions. In these cases, zinc is not released under normal circumstances. Other proteins revealed rapid association and dissociation rates for zinc, short enough for zinc to regulate processes such as pathogen sensing, intracellular transportation and signaling. This pool of zinc is called free, labile, or mobile zinc, describing loosely bound or unbound ions that are thermodynamically available for cellular metabolism. Coordination chemistry of those mobile zinc ions has been barely explored, 30. Thus, it is difficult to create artificial substances that can deliver zinc or chelate it away from protein. This explains why there is so far no clear answer to the question, which zinc compounds are best to use for zinc supplementation. In addition, it underlines why there is still no reliable method to measure zinc status of individuals and the immense discrepancies between studies, even if experimental setups seem to only differ slightly. Zinc is released from bound protein during regular protein turnover. It is mobilized from protein by chemical processes, and can be mobilized between the extracellular and intracellular, organellar or vesicular compartments, 30. Efficient zinc buffering inside the cells, to avoid zinc intoxication and zinc deficiency, is regulated by coordinating expression of zinc-binding proteins and zinc transporters, 37. This includes so-called muffling, where proteins transiently bind zinc and transport it to vesicles, 29. To be correct, Zinc ions are never completely free inside the cell but always complexed with amino acids, phosphates or other low molecular weight ligands. However, free zinc will be used here to distinguish the pool of zinc that is available for signaling from zinc stably bound to protein and therefore unavailable. This is the term generally used in literature in this field, although it is not a satisfactory definition in the chemical sense. 30. After stimulation of cells, intracellular zinc fluctuation, or in short, zinc flux, has been observed, suggesting a function for zinc as a second messenger. 93, 94, 95. In myeloid cell lines, primary monocytes and granulocytes, a zinc flux can be induced by lipopolysaccharide, LPS, or phorbol 12 myristate 13 acetate, PMA, which was not detected in lymphoid cells. 83, 94. Furthermore, Fc epsilon receptor I, Fc epsilon Re, activation caused a zinc flux in mast cells. 96. Increased intracellular zinc in monocytes was also detected after stimulation with monocyte chemoattractant protein, MCP1, tumor necrosis factor, TNF, alpha, insulin and PAM3CSK4. 97. Using various ligands for toll-like receptors, TLR, including PAM3CSK4, TLR1/2, Listeria monocytogenes, TLR2, flagellin, TLR5, FSL1, TLR6/2, ssRNA40, TLR7, and inhibitory oligonucleotides, ODN, 1826, TLR9, all increased intracellular zinc in murine macrophages and primary human monocytes. 94, 97. In these cases, zinc was mostly shown to be increased, but a decrease might occur as well. Zinc can transduce the extracellular stimulus into an intracellular signaling event. Release of zinc from the endoplasmic reticulum has been shown to be inducible by some hormones, similarly to what has been described for calcium. 98. Another source of zinc is zinc-binding proteins, as already indicated. Here, MTs play a decisive role, as they bind up to 7 zinc ions, which can be released rapidly. Zinc ions can be released from their coordination environment with sulfur donors. Zinc is released from cysteine in proteins, suggesting that a redox signal can be translated into a zinc signal. 99, which we will return to later in this review.